Starting this experiment, there were 4 separate glasses, each filled with 5 grams of dry active yeast. 3 were filled with 6 grams of table sugar, and 200 milliliters of water was added to them all, 3 of them being at different temperatures. One glass included yeast, sugar, and ice-cold water. The next one had yeast, sugar, and boiling water. The third one had yeast, sugar, and 37-degree water. And finally, the last one had yeast and 37 degrees water. After a total of 30 minutes, each glass performed a different reaction. Starting with the glass of ice-cold water, after the first 15 minutes, the yeast was not completely dissolved, leaving a slimy/sludgy mixture on the bottom of the glass. There was no rising of any bubbles or foam, and the cold temperature from the glass affected how quickly carbon dioxide would have been released from the reaction. The ice-cold water is too cold for the yeast to consume the sugars quickly, leaving it to slowly produce carbon dioxide and other molecules. Even after another 15 minutes, the results of the ice-water glass remained the same. Since the production of CO2 was slower, that resulted in little to no bubbles and foaming, and not all the yeast and sugar being dissolved because there was less heat and energy to speed up the dissolving of the sugar and yeast. For the boiling water glass, after 15 minutes, the yeast was completely dissolved and had the same consistency throughout the liquid. There were no bubbles or foam or rising in this reaction. Although ice water isn't the best temperature for yeast fermentation, boiling water also isn't. All the sugar and yeast did dissolve because since the water was heated, energy was gained to make the yeast and sugar dissolve faster and easier, but still, only a little CO2 was released. In this glass, the water was too hot that the temperature denatured the yeast enzymes, killing the yeast. The active site begins to change, and once the temperature is too hot, making the yeast unable to rise and release/produce carbon dioxide. Even after the second 15 minutes, the boiling water glass began to develop a scent of burnt crust or dough, which resonates with the thought of the enzyme being burnt due to the boiling water. Next was the 37-degree water with sugar. This water temperature produced the most significant results. The water was a perfect temperature for the sugar to feed on the start of the production of alcohol fermentation. The yeast begins to feed on the sugar as the warm water activates the yeast. This 37-degree water is great for recycling NAD+. We take the product from our first step in cellular respiration, glycolysis, which is 2-pyruvate, and convert it into 2-acetaldehyde. While doing that, we release CO2 from 2-acetaldehyde and convert it into 2-ethanol, which is alcohol. Ethanol gets produced from NADH, giving up a proton and an electron, turning it into NAD+ to use for energy. Alcohol fermentation ends up leaving a high concentration of ethanol that gets removed as it is poisonous, and this reaction doesn't require ethanol and only performs alcoholic fermentation for the recycling of NAD+. This is specifically great for yeast as it releases and produces CO2, which is great for making the dough rise. After 15 minutes, the results of the 37-degree water with sugar were that there were lots of bubbling and foaming on top of the yeast and water mixture, and the foaming reached over 200 milliliters. There was approximately 4.5 centimeters of rising and a sharp, tangy, almost beer-like odor, which resonates with this being an alcohol fermentation reaction. After another 15 minutes, the foam bubbles almost started to levitate over the yeast and water mixture from all the CO2 production. There was now 6.9 centimeters of rising, and the beer-like sour odor remained the same. Finally, the 37-degree water with no sugar was left. After the first 15 minutes, this glass had very little foaming and still some undissolved yeast floating through the mixture. After the second 15 minutes, the yeast and water mixture separated. There were a few bubbles and foam and only 0.3 centimeters of rising. This reaction didn't have a significant appearance of a chemical change due to there only being no sugar for the yeast to feed off of. Yeast needs a supply of energy to live and function to its best ability, and sugar is a great source of energy. The sugar assists with the yeast in gaining energy for it to grow at a faster rate for it to release ATP with the help of oxygen. But since there was no sugar in the glass, the yeast didn't have much energy to begin with, leaving it not to release CO2 on the other glasses if it needed help, leaving it not to release CO2 or other gases needed in helping the yeast rise/ferment. The lack of sugar and energy can eventually lead to the yeast to death. An error made during this experiment was that after the first 15 minutes, all 4 glasses were slightly mixed again before making the second observation. This affected the lab as restirring the glasses left each reoxygenated and losing some of the yeast's ability to ferment effectively during fermentation. The result is a delay in the startup of fermentation. The addition of extra oxygen can alter the production of other gases like CO2, which is needed in fermentation. If we observe these glasses for a longer amount of time, the reactions could alter. The ice water's already slow fermentation could be disturbed by the stirring, but the stirring might have added heat and energy, potentially speeding up the reaction. The hot water glass was mainly hopeless, so excess stirring didn't change the mixture as much. Stirring the 37-degree water with sugar ruins all the CO2 produced and released by the addition of excess oxygen, which makes the yeast lose some of its ability to ferment more. Like the boiling water, the 37-degree water without sugar didn't have much of a change from its original state. Warm water should be enough to activate the yeast and doesn't require excess mechanical energy. Overall, all the glasses undergo aerobic respiration as it uses oxygen, and three of them use sugar to produce energy and CO2. Anaerobic respiration would only occur when oxygen is not present, but all these glasses were open and exposed to oxygen around them. Yeast cells use glucose and oxygen to produce energy, and yeast cells only do it for the sake of producing energy, which results in this experiment undergoing aerobic respiration. In conclusion, throughout this experiment, the recycling of NAD+ was done throughout 30 minutes of the 37-degree water with sugar to produce ethanol from alcohol fermentation. From all the glasses having either sugar and/or yeast, and all of them being exposed to oxygen, it is determined that this lab undergoes aerobic respiration. The different ingredients, whether it was sugar or yeast, and the different temperatures of water, played a significant role in how all these glasses reacted differently to cellular respiration.